Welcome back, everyone, to the Genome Application Symposium. So our next session will be on genome applications for species of economic value. And this session will be chaired by Leif Anderson and Piergiorgio Massa. So the floor is yours.
All right, let's get started. Shall I share my screen?
Yes.
Looks fine. Good. Okay, so we will talk about genome applications for species of economic value. And so I'm Leif Anderson from Uppsala, and then we have Piergiorgio Massa from University of Bologna, Italy. So this is the plan, that we will have 3 short talks by myself and Piergiorgio, and then from an invited guest, Francesca Bertolini, who will talk about animal breeding sector. And then we have a general discussion after that. We decided to skip the Mentimeter and have more normal discussion. So early this year, we published a review paper in Annual Review in Animal Biosciences, where we called a roadmap for how fish population genomics can promote sustainable fisheries. So this is one of the figures in that review. So one of the arguments there is that, so this is about marine fishes. So the fishes that is exploited by commercial fishing are abundant species, often huge population sizes, which means that there's very little genetic drift. There's often gene flow between subpopulations. And that makes a strong argument to have a high quality assembly to really find the loci that is most important for differentiation between populations. So this is what BGE is doing, creating high quality assemblies. And the next step in the application of this method is to identify informative genetic markers. And that is done by looking at baseline populations. So you sample the species from where spawning occurs and try to cover the populations as good as possible. And then in our case, we have been doing whole genome sequencing to identify the loci that show genetic differentiation between populations. In the next phase, we design a genotype method for genotyping, so which can be applied on a large scale on thousands of fish. And then you establish those genetic markers that can distinguish the different populations that you are familiar with. And in the fourth case, this is applied then to fisheries management, where you often have mixed samples of mixed populations, and you would like to assign the samples you have taken from a certain area to a certain stock and be able to judge how the different subpopulations are doing. And I have been working on Atlantic herring. I call this a gold mine also for exploring the genetics of ecological adaptation, and I will come back to that. So the census population size is about 10 to the 12. So you could wonder, why do we need to be concerned about the conservation of one of the most abundant species on Earth? But the thing is that there is biodiversity also in a common species like this. So this is from a paper we published a couple of years ago, where we described the major ecotypes of Atlantic herring. So we have, from the Atlantic Ocean, we could split them into autumn and spring spawners, which are genetically differentiated. This is the populations around Ireland and Britain. The transition zone is this, when you go from the Atlantic into the brackish Baltic Sea. This is the Baltic Sea population, also split into spring and autumn spawners. And these were classified as a subspecies. These are all classified as Atlantic herring. And these were classified as a subspecies by Linnaeus in the 18th century. So these are what we consider the major ecotypes of Atlantic herring. And for instance, in this area, the North Sea, many of these populations are mixing for feeding here, and there is a very extensive fishery going on here. And when stock assessment is made, you would like to assign fish to the correct stock. So this research has developed into a genetic tool for stock assessment. So we have developed what we call a multi-fish SNP chip, 7 different species, so 4,500 SNPs in Atlantic herring. And these are now being picking up by the stakeholders for stock assessment of Atlantic herring in the Northern Atlantic. So I wouldn't be surprised that for the next year, there will be maybe 20,000 fish or so genotype using this SNP chip because it is the most efficient way to characterize herring stocks at the moment. But I would like to take up another example of diversity that we was not aware of. So this is what I showed you in the previous picture was the broad picture. So here we decided to go only into this small area of the Baltic Sea and ask the question, what type of subpopulations do we have there? And interesting, that led to the discovery of evolution of piscivorous Baltic herring, which is a very surprising discovery because herring is an iconic planktivorous fish, which is super important for the ecosystem of the North Atlantic, is the link between plankton production and higher trophic levels. And until recently, we thought that herring always was planktivorous, but we discovered something what has the name in Swedish, slåttersill. And I discovered this because of I have a summer house at the coast, and my neighbor there is a local fisherman. And he told me that every year around midsummer, there is a type of herring coming in, which they call slåttersill. And then I need to teach you some Swedish here. So slåtter means hay harvest. So these are spawning around hay harvest every year. And sill, that is the Swedish name for the Atlantic herring, the other subspecies. And Atlantic are larger and more fat than Baltic herring, and that is the case for these as well. So what is this? So this advertisement we discovered, which is an advertisement for where slåttersill was on sale for an advertisement from 1903. So apparently this herring has been known by the locals for hundreds of years, but has not really been described in the scientific literature. So if you look at the growth curve, if you measure age, so this is the small planktivorous herring, spring and autumn spawners, and this is this slåttersill, which has a much higher growth rate. If you do SNP chip genotyping with this tool that I described, then you see this is the autumn spawning planktivorous, this is the spring spawning planktivorous, and this is the, you could say, late spring piscivorous herring. If you then do GWAS comparing this piscivorous herring from other populations, what you see is quite interesting is that you see very significant genetic differentiation at certain loci with p-values 10 to the minus 40. There's no question that these are genetically differentiated, but you could see for most chromosomes, there's very little signal. So we don't see differentiation at neutral markers. And we think this is because there is gene flow even between the different ecotypes. And if you go to population genetics theory, you know that very little gene flow on the order of 1 migrant per generation is sufficient to avoid differentiation at neutral loci. And this is the reason why it is a gold mine that when we see difference in allele frequency, it's because of natural selection of active. If you then look at, this is fat content. So this is this piscivorous herring. This is the planktivorous. You see a big difference in fat content. This is the dioxin pollutant, which is a major issue in the Baltic Sea, that we have a pollutant present. And And we had expected that this piscivorous, because it's more fat, should have higher pollutant, but it has significant lower, which means that it's better suited for human consumption. And this result implied that they have an altered feeding or migratory behavior. An interesting aspect, if you look at the gill rakers, so this is the gill rakers of normal planktivorous herring. And they use these gill rakers to sieve plankton. This is from this piscivorous herring, and you see the damage here, because we think an important feed for this are sticklebacks, which has big spines that is difficult to consume for this fish. Then after that, we did whole genome sequence, because the SNP chip contained a lot of SNPs on the selection, but not all. And we decided to do whole genome sequencing of this piscivorous herring, and together with some other unusual large herring from other parts of the Baltic Sea. And there was evidence for a fish diet in this other large herring based on stomach content. So, and what this resulted in is that we identified that there is at least 2 subpopulations of piscivorous Baltic herring. We call them the northern piscivorous Baltic herring. So we find that in this area here. So this is the island of Åland. This is the Bothnian Sea. And here is where we find the northern piscivorous. The southern piscivorous is found from Stockholm and south of Stockholm. In this area, we find the southern piscivorous sea. And this is the average FST between them. And you could see that FST values between, so this is northern piscivorous, southern piscivorous, spring planktivorous, autumn planktivorous. And you see the FST between them is on the range 1.6 to 2.6% among this. So they're very, that is because there is basically no genetic differentiation at neutral markers. And interestingly, if you do a phylogenetic tree, we see that the northern piscivorous cluster with, the northern piscivorous clusters with the spring spawning planktivorous rather than this is the, let me see, so this is the northern, southern piscivorous. And we think, when we look at the signals of selection, we still think that the northern and southern piscivorous has a common origin. But since they've been geographically separated and they are surrounded by billions of planktivorous, there is some gene flow between them. So for that reason, could even be some climatic adaptation, that the sea is a bit colder up here than it is down here, which could also lead to this type of changes. So that is so there's apparently gene flow going on between them. So the important question is, of course, why has a fish-eating ecotype of herring evolved in the Baltic Sea but not in the Atlantic Ocean? And then one should remember that the Baltic Sea is a young water body. It is around 8,000 years old. So after the last glaciation, colonization happened, and there's a shortage of marine fish in the Baltic Sea. So for instance, we don't have the mackerel, which is in fact one of the species that we work in our case study in BGE. And so that means that there is empty niche space for a piscivorous species. And we think this has been the opportunity that led to this evolution of this piscivorous Baltic herring. So I think this is a really interesting example. You know, you could say that if you would look at herring is the least concerned with regard to conservation genetics, because it is one of the most abundant vertebrates on Earth. But what this shows is that there could be very important biodiversity within these common species. So so for instance, this piscivorous Baltic herring can be really important for the ecosystem because it fills an empty space in the ecosystem in the Baltic Sea. And I would argue that this is a ecotype of herring that has a very high value for be maintained as a biodiversity resource. And you know, the traditional way to looking at how much nucleotide diversity is existing as would never have identified this as a problem, because this will, but so I think the message from this is that even in these common species, it's really important to understand population structure to decide on how to maintain biodiversity in nature. That was all. Yeah. So I think I could skip this. This is why herring has such a remarkable population structure. I think we go to the next, which is Piergiorgio, that will talk about from ancient genomes to modern conservation, an Atlantic bluefin tuna case study. So I stop share.
Hello everyone. I'm sorry, my PC camera decided to stop this morning, so I have to use my mobile phone camera. I hope this is not a problem. Okay, I'm going to share my screen. Okay. Okay. Can you see it?
Yes.
Yes. Okay. So, yes, I'm Piergiorgio Massa. I'm a PhD student from the University of Bologna. My PhD thesis is on applying genomics to Mediterranean fisheries. These three here are the fish species on which I'm working on currently. But today I'm going to talk to you about the bluefin tuna, Tunus tinus. Indeed, very recently I started this project on the adaptation and conservation genomics of this species, granted by ERGA-BG, and in collaboration with a team of researchers from University of Bologna, Polytechnic, University of Marche, Norwegian Institute for Water Research, and Norwegian University of Life Sciences. And this project starts from the work of Adam John Andrews. He is a he has been a PhD student in our lab in Bologna and is now based at the Norwegian Institute for Water Research in Oslo. For his PhD, he has carried out huge multidisciplinary work on the exploitation history of Atlantic bluefin tuna. He has done literature review, but also isotopic, morphological, and genetic genomic analysis on both modern and ancient samples. And why the bluefin tuna? Because first of all, because of its huge economic but also sociocultural importance, and also because it has a very long exploitation history that started some centuries before Christ. Then it has increased quite a lot some centuries ago, as it can be seen by the increased number of tuna traps across the Mediterranean area. And then it has further increased, reaching overfishing levels that made it necessary to set fishing quotas to make the population recovery. So for his PhD thesis, he has done also genomic stuff. And he has not, this is the work, but is still under review, but you can find it as a preprint with this QR code here. He has produced this whole genome dataset of 90 samples from both the western side in blue, from the Gulf of Mexico, of the Atlantic Ocean, and from the eastern side, from the Mediterranean, the whole Mediterranean Sea, and from Norwegian seas. And this dataset can count also on ancient samples, 41 ancient samples from 1941 to 3000 before Christ. From this dataset, he first confirmed the two-stock structure of such species with a western and a eastern stock with high connectivity levels, as is commonly observed for large marine migratory fish species. But then he also managed to find a decline in the effective population size of the species that started concurrently with the starting of the industrial scale exploitation. Then he also compared genetic diversity estimates of ancient and modern samples, and he found a decreased nucleotide diversity and an increased observed homozygosity in the modern samples compared to the ancient ones, as expected by after the observed bottleneck, the observed decline. And what is important to notice is that the quotas, the abundance base, the 1970 abundance baseline that have been used by ICAT to set the fishing quotas for such species have been set according to an already altered state of such species. And this is a phenomenon that has been already described by Pauli in the 1995 as the shifting baseline concept, whose result is the gradual accommodation of the disappearance of research species, as he already stated. And now let's get back to my project, which comes as a continuation, integration of the Adam's work. And what our goal is to resequence the Adam samples to further increase their coverage, since he managed to obtain the around 10x coverage for both modern and ancient samples, and while we aim at 20x. And this, together with a new high quality chromosome level reference genome that has been very recently produced, we aim at study focus on the non-neutral genetic variation of such species. And these two are the two research topics on which we want to focus on. I know that this will be definitely challenging because of the genomic stability that has been already observed in other commercially exploited marine fish species like the Atlantic cod, because these FIE traits have been already explained by phenotypic plasticity, because it has been already hypothesized the recovery of former genotypes concurrent with demographic recovery, and because it can be hard to disentangle the source of the evolution if viscera-induced or climate-induced. And then as for genetic load, again, the genomic stability can cause the genetic load to stay still across time, but this is not what we expect, since Adam already observed a decline in the neutral genetic variation. And yeah, because the genetic load is poorly investigated marine fish species, and because usually it is for species with smaller population sizes. But yeah, that's why we have research partners. So let's see what we get. Now let's leave the floor to Francesca. No? Okay.
Can you see the screen? Can you hear me?
Yes.
Okay, so good morning. I'm Francesca Bertolini, Associate Professor at the University of Bologna, and I will briefly talk about some example on how genomic-based competencies can be nested into bigger pictures to, I would say, solve or address complex themes linked to animal breeding and aquaculture. Sorry, I cannot. Okay, sorry. Just to give to try to find a common ground of the three initiatives I will present, I think that it is important. I mean, it's obvious to notice that, of course, genetics and genomics of animal species are fundamental. We are all here. We all work with genomics, and it is the background for basically everything. However, there are also other the genetic and genomics can be interact, and it's a good fact that it interacts with other disciplines, and also genomics can be declined in a little bit different ways. We have genomic of the animals. We've seen a lot of nice projects from my colleagues before me, where it's the animal genetic code, and of course it's also important for any kind of selective breeding schemes if we were tackling animals with economical relevance that can be bred in captivity. But then we have also functional genomics, so how the genetic information is expressed, and also the way that they can be used as a biomarker to monitor the physiological conditions and stages of any kind of animals in any situations. And then there is also environmental genomics that goes beyond a little bit environmental DNA, but where environmental DNA of pathogens or microorganisms can be used can be linked to the genome of the host and analyze a bigger picture in a more holistic way where an animal is living. And this is particularly true, I would say, for aquatic animals that are raised, so they live in a tank where the concentration of microorganisms are for sure higher than a livestock that lives in a farm where DNA of the hair has been collected as research are going on, but they are not as much as informative as the environmental DNA that can have provide from the water. For this, I will bring you three examples of coast actions that are which I am involved that are targeting these aspects in different ways with different purposes, and we will give us a few keywords. So it will be ill support, it will be bioaqua, and it will be Leif. But first of all, one definition of what is a coast action. Coast action is a funding initiative that is provided by the European Union, where basically it does not support any research activity as such, but it provides money for connections, for short-term scientific missions, so to dig into several problems in a complex way. That normally is also the baseline nowadays to then develop more complex projects. They are bottom-up. That means that every idea that is valuable can be developed. And basically, it is a baseline where multidisciplinary aspects can be gathered together and expertises as well to answer particular questions. So the first example I'm bringing to you is a very targeted example, and it is focused on the European eel. The European eel is a very important species for European food system. Is a niche, but it's a traditional product. It is also, in my opinion, a very fascinating and mysterious animal with still a lot of missing spots in the wildlife. And it isn't critically endangered. So that's why it is fundamental to close the life cycle and be raised in captivity. Now, the main goal is not to use genetic at this moment. The main goal is to close the life cycle. Of course, after this will be closed, breeding programs will be started, as is starting already with the Japanese eel, and then genomics can be fully utilized in the way that animal breeding and aquaculture is doing. But this cost action is have 4 work packages, and they are really linked to the different steps that are important to close the life cycle. But I want to focus on the contribution that for example, transcriptomics can give and is giving to the first 2 work packages, root scar conditioning and sexual maturation. In fact, we have shown that blood transcriptomics from whole blood transcriptomics, at least a little bit of blood, not that much, combined with machine learning, is able to answer and discriminate among different questions, both at ecological level, so to understand better the behavior of migration of eels, to discriminate between animals that are shifting between habitats and animals that are stationary in same habitat, and also can be applied with aquaculture. In fact, we have seen the blood as a powerful discriminant between good spawner and bad spawner that will be used probably to reduce to optimize the very difficult vitellogenesis and maturation process that is very expensive nowadays, and therefore knowing in advance in a good spawner, it will be a good spawner or a bad spawner, will be very important, have an economical impact. And then we move to another topic, another question, broader, is still aquaculture, but it is linked to the disease and management practices, basically of all species that can be raised. The goal is broader, providing technologies for integrated approaches in biosecurity, having healthier animals, and removal and identification of pathogens, and so on. What is relevant to genetics and genomics is relevant here is, for example, in work package, working group 2, biomolecular solutions, where I'm co-involved, when we are trying to gather all information as possible to see if and how far we are to develop combining sequencing technology combined with artificial intelligence for developing in situ and routine identification of pathogens in the water in aquaculture facilities and companies. So that will be also then the platform to then maybe link this pathogen to the genome of the host in a second step. And then the last example I want to bring is it's a more complex, is target livestock, but not only, because it's also applied to aquaculture. In fact, we have a lot of aquaculture, more and more aquaculture research that are joining, is Leif, livestock phenomics. Because in animal breeding, we can say that I don't say what we have genomics has done everything. It's not true, but we have a lot of different tools. We also speak about pangenomics and so on. What we are missing are phenotypes. And therefore, to make the best use of genomic technology, we need more phenotypes. And those phenotypes can be internal phenotypes. We talked about transcriptomics as biomarkers. We can talk about metabolomics and so on, but we need more the use of advanced technologies such as sensors that can monitor in real-time an animal behavior and health and so on. And so this cost action is focusing, trying to combine and optimize and collect all this information. And this cost action is divided in 5 work packages, where you can see the work package 2, where I know a little bit more. It is really the integration of all the phenotypes that are collected in group 1. So all the phenotyping technology with the genomic information that we have. We talk about the majority of livestock, where the genome is very well known, well annotated, and resources are a lot, but genomic resources, I mean. But also aquaculture is joining that, because a lot of technologies to predict the yield of a fish and so on, of the quality of the meat of the fish before it is slaughtered, it is becoming more and more important. And then, of course, a lot of information need dedicated computational resources, and then, of course, also to talk with policymakers and stakeholders. So concluding, genomics is key element for any kind of species, animal breeding and management. Of course, we have a high array of genomic tools and integration, and these tools need to be integrated with other disciplines in order to take the maximum out of the information that an animal genome can give to us. And for example, cost action can be an ideal platform to begin this integration then for future projects that can be led from these initiatives. And I thank you for the attention.
Thank you very much. So we have 10 minutes so maybe we could start if there are questions on the 3 presentations. Yeah, one, Jaakko.
Oh, hi, let's start. All 3 fascinating. I was actually wanting to get back as a Finn to your introduction to the Baltic herrings.
Yeah.
So to my knowledge, there used to be kind of endemic larger herring in the Baltic Sea, but that went extinct somewhere in the Middle Ages. Is it possible that your herrings are some sort of relict population of those?
I mean, we definitely think so that this has a long history, this large herring, and they still exist. Apparently, what I maybe didn't say is that they are rare. So I don't think they constitute more than 1% of the herring population and I guess they're very vulnerable to fishing because they're big and they are catched in trawls and during trawling and netting. And I know that there are, in fact, I have got samples from big herring from Åbo archipelago in Finland.
Right.
That we will test and see how it fits with this model. So
right, but there's no evidence that they would be kind of left over of the older Atlantic.
I think we can't tell yet. I mean, what would be interesting would be to look at old samples, and I mean there are collections of fish from and see if
surströmming stocks.
Yeah, surströmming is definitely done with these small planktivorous fish to get them into the box. You know, these guys otherwise are too big to get them in there. So
right. Okay, thanks.
Thank you. Any other question?
There should be a question in the chat, I think.
I didn't see any, but
I think it's for Francesca.
Okay.
Which phenotype ontology are you planning to use?
Probably technology. Maybe it's I guess if it's the question. I guess ontology, I don't know, but I think technology. I mean, we are collecting any kind of phenotypic technology that can be utilized. For example, if you talk about livestock sensors that are recording milk, temperature of the animal, the heartbeat, everything, this is more for a cow, but for fish, for example, every technology that are now developed to scan, to predict the quality of the fish meat before slaughtering, or sensor that are monitoring the status and some like components in the water and so on. So phenotypic technologies are can be several, and also internal phenotypes. Metabolomics, for example, we are doing in our group a lot of work on metabolomics in pigs and cattle, and hopefully in fish, to then be combined into genomics and calculate heritability of those traits. So where genomic is always nested into all these phenotypes.
So a comment on your presentation there. So you didn't mention that, but in domestic animals, genomic selection is widely used these days, which is based on whole genome sequencing and large-scale SNP genotyping to predict EBVs, estimated breeding values. And that is the major method to used in pig breeding, cattle breeding, chicken breeding, I would say, probably in aquaculture as well. So that's very obvious how genomics contribute to that. So let me see. I try to share screen. So how can genomics improve the management of species of economic value? So I think it's quite obvious from the presentation you heard that genomics are very important in these species, and I would like to make a comparison between the Atlantic herring and the tuna presentation that we did. So what we decided to do, so in fact, this overview I show with the ecotype, major ecotypes of Atlantic herring, we did it by pooled sequencing. And we did that in order to be able to get data from many thousands of fish. And you know, whole genome sequence is still expensive when you do it on thousands of individuals. So we decided to do pool sequencing. So what we started with was to sample fish at the spawning site, because with the assumption that when they are spawning at the same site, they most likely belong to the same population. And then included a worldwide distribution. So basically we have samples from essentially all the species distribution, and that is how we could do that. But I noticed in the tuna project, there was quite limited number of samples that have been collected. And now you plan to use the same samples and increase coverage. So my question is about the tuna presentation is, so how many samples have been sequenced so far, and do you have plans to expand? So it looked like you have two populations, a Western and the Eastern, but is there a risk that you're missing heterogeneity within those two major groups?
Yeah, so the point is that when we talk about ancient samples, one point is that it can be hard to find some specimens from a specific time. I have to admit that what I talked about in my presentation is the project of Adam John Andrews. So he has gone through the, let's say, the preliminary part where the cost analysis, where you carry out the cost analysis to check how many samples can we get, where we can get the samples. So I don't know, I cannot share these results with you, but yeah, what I can say is that when we work with ancient samples, is trying to get these can be hard. So at this moment, what we want to do is to perform adaptation analysis. Our budget is limited, so what we decided to do is just to make these samples at a higher coverage and not to produce other samples rather than produce other samples with a low coverage, because it is better to have hardly called genotypes when you work with adaptation analysis. So yeah,
Right.
but and since I'm at the I'm starting the third and last year of my PhD, it would have been hard to collect some other samples for this project. So yeah, this is a way safer way of going on with the project. Yeah.
So I give the word to Michele Cassina, which is in fact have been working with stock assessment for a number of years in ISIS and so on. So I'm looking forward to hear your comments.
Thank you, Leif. Nice to meet you again after some time. So I just wanted to make two comments. So you wrote here these questions, but I can make an example that there is another species like the European sprat, which is mixing with herring in the North Sea and in the Baltic Sea. And actually there, in this case, genetics have, for example, confirmed the stock separation that we were using in stock assessment and management. So this was very important thing, because there were many doubts about the separation of the stocks as we treated them in the assessment. So by using the genetic tools, we were able to confirm the separation. So for us was very good to know that we were not doing a bad job, let's say, in this sense. And then I was very interested about your story about slaughter sill.
Yeah.
That is a big question currently in Sweden. And I know that there are many big projects trying to understand why the big fish along the coastal waters have disappeared in the last years. And this has to do with also the possibility to move the fishing line more far away from the coast.
Yeah.
And then of course this depends very much on the fact if these big seal are different genetically compared to the other one. So I think of course these genetic tools can give the question about this. So they will have a big impact on the management along the Swedish coast and also Finnish or other coastals. So of course is extremely important. These are two comments. Thank you, Leif.
Thank you, Michele. I agree that I think our new discovery is a really important argument to reduce extensive trawling close to the coast to maintain this larger herring. So we hope that is so. The paper is currently in revision for Nature Communications, and we plan to send out press releases, and that will be one of our arguments that this need to be taken into account in stock assessment in the Baltic Sea. Right. So Jose.
Yes, Leif, I have a question about your SNP ship. So you mentioned that you expect it soon to have been used to genotype 20,000 individuals, right, for stock assessment. But how did it work in the beginning? So did you produce the SNP ship as a sort of a demand from the stakeholders, or did you produce it just for scientific reasons and then it was picked out by them?
So the story was that there was a Swedish authorities for management of the oceans or sea, and they wanted to monitor biodiversity in Sweden, and they decided to have a number of species that were like key species to look at, and they selected both plants and animals, and herring was selected as one of the key species to monitor, and they provided a grant to do genotyping of something like 5,000 herring from the Swedish waters. And then they contacted me about how to do that and so on. And the initial plan was to do 100 SNPs and so on. But we convinced them that it's better to try to develop another method that is more cost effective, generating more data. And then we came to the idea that we could do, the problem with SNP chip is fantastic. That is how all jivas in humans is based on the SNP chip. But the thing is that it's costly to make them to start with. But if you could increase the volume, and the idea was to add multiple species onto this SNP chip so that it could be used for monitoring. So then we came into an agreement with the company to develop this SNP chip with 7 different species. So within BG, we have 2 fish species that we're working on, the mackerel and blue whiting, and the libraries have been made for mackerel now. So we will have, I hope we will have result before this year for both species. And the idea is to include them on the new version of the SNP chip that we will start discussing in January. So the idea is to those species where there is an interest to do stock assessment, they can be included on this SNP chip, because 4,500 is in fact more than you need in this case. But they are fantastic as a research tool, I would say. I think we are running out of time, right?
Yeah.
Maybe we could take this because this was one of the questions we took. What are the challenges in applying genomics to economic important species? I think we could take herring as an example. There was a recommendation from the European Commission to stop trawling in the Baltic Sea, but in the European Parliament they didn't follow the recommendation from the Commission. So the fishery biologists argued that the reduction of this herring stocks in the Baltic is too dramatic to allow trawling. But then what happened in the parliament was there were these lobbying activities from economic interest from, I don't know exactly who did it, but among those that, so the herring from the Baltic Sea is used for fish meal production to feed salmon and other animals. So so that is the, you know, the knowledge is there that there have been too heavy pressure on this population. But in order for genomics to have an impact, that they need to be taken into account when the fishing quotas is set. And there is this conflict between biology and economic interest. I'm sure Michele has a lot of experience of that.
Well, I mean, this has been always true. I mean, we give a scientific advice, but our advice is based only on biology, let's say. And then the parliament needs to take account of, you know, cultural, economic, social interests. So it's difficult to get through, let's say. Yeah.
Right. Thank you very much, everyone.
Okay, thank you, Leif and Piergiorgio, and also Francesca for your presentations and discussion. So we will now stop for lunch at the symposium, and we will come back at 2 p.m. CEST. See you later.